Good morning, students. In the last lecture, we were discussing about the ultrastructure of the bacteria. In the theory examination, the short note may be asked on the ultrastructure of the bacteria. Okay, the ultrastructure of the bacteria, the cytological details of the bacteria, these can be only best studied under the electron microscope and also using certain staining technique. Okay, in the last lecture we have discussed about that the bacterial cell, it is a typical prokaryotic structure. Okay, bacterial cell, it is a typical prokaryotic structure, and here the bacterial cell, it is surrounded by, it is enveloped by the three layers. The outer one, it is called as the capsule. In some of the bacteria, it is also referred as the slime layer. Okay, the function of the capsule or the slime layer is to give the protection, that is, these are protecting the bacterial cell from the desiccation and from the antibodies. Then just below the capsule wall, there is a presence of the cell wall. Okay, just below the capsule there is a presence of the cell wall. It is the middle layer. The function of the cell wall, it is truly mechanical, because it is giving the shape and rigidity to the bacterial cell. Here the cell wall, it is completely different from the cell wall which is present in the higher plant. It is not made up of cellulose. Okay, we have discussed regarding the cell wall in the last lecture. Then just below the cell wall, there is a presence of the plasma membrane. It is the innermost layer. It is called as the plasma membrane. It is also referred as the cytoplasmic membrane. And plasma membrane or the cytoplasmic membrane, it is the center of the multifarious activities, because it is secreting the different types of the enzymes which are uh, very important, which are playing an very important role in the different metabolic pathways. And also the cytoplasmic membrane or the plasma membrane, it is controlling the entry of the organic and the inorganic compounds in the bacterial cell. Okay, then also we have discussed about the cytoplasm and the inclusions. Okay, cytoplasm and inclusions. The cytoplasm, it is a liquid which is having the it is having the different types of the complex organic compounds. Okay, and also in this, it, as it is a typical prokaryotic cell, there is a complete absence of the well developed nucleus. Then the cell organelles like the mitochondria, then the endoplasmic reticulum, then the plastid like the chloroplast, these are entirely absent in the bacterial cell. It is a typical prokaryotic cell. In the photograph, clearly you can see there is a complete absence of the well developed nucleus. Well developed nucleus means the structure of the nucleus, it is having the nuclear membrane. Then in the nuclear membrane, the nuclear pores are present, the nuclear sap is present, nucleolus is present, and in the nucleus then there is a presence of the chromonemata. Okay, but here you can see the simply the genetic material which is present in the center region. Okay, there is a complete absence of the well developed nucleus in the bacterial cell. Okay, then here you can see the instead of mitochondria, there is a presence of the mesosomes. The function of the mitochondria, it is performed by the mesosomes. Okay, then also we have discussed regarding the structure of the genetic material. Okay, here the genetic material which is present, it is maybe double stranded or the single stranded DNA. Okay, then it is here you can see it is called as the region where the nucleic acid it is present. That region, it is called as the nucleoid or the genophore. Okay, and generally the double stranded DNA in the bacterial cell, it is called as the bacterial chromosome. Okay, and this bacterial chromosome, it cannot be compared with the chromosome of the eukaryotic organisms, because there is a complete absence of the histone proteins in the bacterial chromosome. Also then we have discussed about the plasmid. Okay, the plasmid, here you can see these are the extra chromosomal hereditary determinants. Okay, extra chromosomal hereditary determinants. These plasmids possessing, these are nothing but the double stranded DNA. Okay, this is the double stranded DNA, this is circular, and this plasmid certain genes which are not very important. These are not playing an important role in the viability and the growth of the bacterial cell, and that's why the genes which are present in the plasmids, these are called as the non-essential genes. Okay, then the replication of the plasmid, it is completely independent that of bacterial chromosome, and it is having its own replication system. It can replicate on its own, it is having its own system of replication. Okay, then the different types of the plasmid up till now are reported. For example, the F factor, that is the another plasmid which is called as the, which is determining the maleness in the bacteria. Okay, then the another plasmid R factor, R, that is the, these are the plasmids which are showing the resistance. Then col plasmid, col, that is which are having the resistance to the carcinogeny. Then the plasmid, that is these some types of the plasmid, these are responsible for the tumor inducing in the plants. These are responsible for the formation of the tumor in the plants, and that's why such type of the plasmids are called as the tumor inducing plasmids. Okay, apart from this, we have also discussed the classification of bacteria depending upon their staining property. Okay, the staining technique which was discovered by the Christian Graham in the 1884, we have discussed in detail in the last lecture. Okay, as per the Christian Graham, there are two types of bacteria: the gram positive bacteria and the gram negative bacteria. Okay, we have discussed in detail all these factor in the last lecture, and here I am giving some summary of the last lecture. Okay, here I have given the summary of the last lecture, and now we are moving towards the next cytoplasmic inclusion, and that is the ribosome. Okay, and that is the ribosome. Here in the cytoplasm, you can see the large number of the ribosomes, that is these are freely scattered in the cytoplasm. Okay, the ribosomes, these are the important cell organelles because these are the site of the protein synthesis. Ribosomes are the sites of the protein synthesis, and here you can see the ribosomes, the large number of the ribosomes are freely scattered in cytoplasm. Okay, in the eukaryotic organism, the ribosomes, these are mainly attached to the surface of the endoplasmic reticulum. Okay, if you see the eukaryotic cell, then in the eukaryotic cell, these ribosomes, these are attached to the wall of the endoplasmic reticulum, and that's why these reticulum, these endoplasmic reticulums are referred as the rough endoplasmic reticulum. Okay, the endoplasmic reticulum having the ribosome on its wall are referred as the rough endoplasmic reticulum. But as the bacteria, it is a typical prokaryotic cell. In the bacteria, the endoplasmic reticulum is absent, and therefore the large number of the ribosomes, that is these are freely scattered in the bacterial cell. Okay, in the bacteria, these are free, and about 10,000 to 15,000 ribosomes are present per bacterial cell. Okay, about 10,000 to 15,000 ribosomes are present in per bacterial cell. Then the ribosomes, these are different from the eukaryotic cell. These ribosomes are the 75s type. Okay, 75 yes type. Yes means yes, it is a Swedberg unit. Okay, 75 yes, here yes, it is denoting the Swedberg unit. Swedberg, that is Swedberg, gave the classification of ribosomes as per their rate of sedimentation. Okay, as per their rate of sedimentation, the ribosomes are of different type. That means the ribosomes of the eukaryotic organism and the ribosomes of the prokaryotic organism, that is, these are different, their rate of sedimentation is completely different. And here the unit of rate of sedimentation, it is denoted by the capital S. Okay, and is Swedberg, that is the name of the scientist. Okay, Swedberg 75S, that means the Swedberg units, which is determining the rate of sedimentation of the ribosome. Here the ribosomes which are present in the bacterial cell, these are of 75S type. A 75S type, and it is made up of two subunits. Two subunits. Okay, the ribosome in the prokaryote, it is made up of two subunit. Of this, the one subunit is larger and the another subunit is smaller. Okay, the larger subunit, it is of 50S. Okay, the larger subunit, it is of 50S, and which is made up of two RNA molecules and 35 amino acids. Okay, 75S type of ribosome in the bacterial cell, it is made up of two subunits, of which the larger subunit, it is of 50S, which is made of two RNA molecules and the 35 amino acid molecules. Okay, the while the smaller subunit, it is 30S type. Okay, the smaller subunit, it is a 30S type, which is made up of one RNA molecule and about 21 amino acids. Okay, one RNA molecule and 21 amino acids. Here in the photograph, you can see the large number of the ribosomes are scattered freely in the bacterial cell. But here you can see the some of the ribosomes, that is about group 4 to 6 ribosomes, that is, these are present in a group. These are attached with each other with the help of mRNA. Okay, there here you can see the group of 4 to 6 ribosomes which are attached with each other with the mRNA, and in that case this type of group, it is called as the polyribosome. Poly means many. Okay, that means the large number of the ribosomes are freely scattered in the bacterial cell, and sometimes 4 to 5 ribosomes, they are forming the group. These are attached with each other with the help of the mRNA, and such a group is referred as the polyribosome. Okay, such groups are referred as the polyribosomes. Okay, that is all about the ribosomes, and now we are moving to discuss regarding the mesosomes. Okay, here the mesosomes are present. Here the mesosomes in the photograph, you can see these are the complex localized inflorescences of the cytoplasmic membrane. Okay, the mesosomes, these are formed by the complex infoldings, complex localized infoldings of the cytoplasmic membrane. There are about 2 to 4 mesosomes are present per bacterial cell. Okay, there are about 2 to 4 mesosomes are present per bacterial cell. Generally, the number of mesosomes, these are higher in the cell which are having the high respiratory activity. Okay, that means the bacteria which are having the high respiratory activity, in such a bacterial cell, there are higher number of the mesosomes are present. Okay, for example, the nitrifying bacteria. Okay, in the nitrifying bacteria, the number of mesosomes are higher because these are showing higher respiratory activity. Okay, that is the nitrifying bacteria having the higher number of the mesosomes in their cell. Okay, and that's why the some of the scientists believe that the mesosomes, these are the sites of the respiration. That means these are the powerhouses of the bacterial cell, and these are playing the role the mitochondria in the bacteria. Okay, as the number of mesosomes, these are higher in the bacteria which are having the higher respiratory activity. Some scientists believe that these mesosomes are performing the function of the mitochondria. That is, these are the sites of the respiration, and these are the powerhouses of the bacterial cell. But for the but some of the scientists opposed this view, because for the respiration the certain types of the enzymes are required. For example, in the mitochondria, the to perform the whole respiration process, they require the enzyme like the ATPase, then dehydrogenase, and the different types of the cytochromes. Okay, these are present in the mitochondria, but in the mesosomes, the enzyme like the ATPase, then dehydrogenase, and the cytochromes, that is, these are entirely absent in the mesosomes. And that's why there is another view regarding the function of the mesosome, is that mesosome, these are not the sites of the respiration. Okay, these are not the site of the respiration. Okay, respiration, it is nothing but the oxidation. Okay, it is a oxidation process, and for that oxidation the different types of the enzymes are required, like the ATPase, dehydrogenase, and the cytochromes. But in the mesosomes, these are entirely absent. Okay, these are the enzymes like the ATPase and the dehydrogenase, and also it is requiring the certain pigments like the cytochromes. And these cytochromes, then enzyme ATPase and dehydrogenase, these are present in the mitochondria, but the mesosomes are completely devoid of these enzymes and the cytochromes. Okay, that's why some scientists believe that the mesosomes, these are the not the sites of the respiration, but these mesosomes participate in the formation of the transverse septum during the cell division. Okay, that means these mesosomes playing an important role in their reproduction, that is in the binary fission. Okay, binary fission, it is the most common method of asexual reproduction in the bacteria. Okay, and in the binary fission means what? Binary means two, that is from one bacterial cell the two daughter bacterial cells are formed, and in their formation these mesosomes are playing an important role. These are responsible for the formation of the transverse septum during the binary fission. Okay, that is all about the mesosomes. Now here, after the mesosomes, we are going to discuss about the flagella. Okay, all you know that this is the flagella. These are the locomotory organ. Here you can see the two flagella here present, and that these flagella, these are the locomotory organs. That means with the help of the flagella, these bacteria can move from one place to the another place. Okay, the flagella here you can see in the photograph, flagella, or singular, it is called as the flagellum. Okay, the flagella, these are the hair-like appendages. These are hair-like appendages, and these are locomotory organ. Okay, these are hair-like appendages, and these are locomotory organ. Then generally these flagella, these are uniform in their thickness. Flagella, these are uniform in their thickness, and these flagella here in this photograph you can see they arises from the cytoplasm below the cytoplasmic membrane. Okay, these flagella, these are arised below the cytoplasmic membrane from the cytoplasm. They arises from the cytoplasm just below the cytoplasmic membrane. Then these flagella, that is, these are entirely absent in the spherical or the coca shaped bacteria, but these are present in the bacillus or the rod shaped bacteria. Okay, we have discussed regarding these different shapes of the bacteria. The bacillus or the rod shaped bacteria may be motile or may be non-motile. That means some of the rod shaped bacteria possesses the flagella, and some of the rod shaped bacteria, these are not having the flagella. Okay, but the cocci or the spherical bacteria, the flagella are completely absent. The cocci or the spherical bacteria, these are non-motile. Okay, then also these flagella, these are present in the spiral type of the or a helical type of the bacteria. These are also present in the helical type of the bacteria, then the vibrious bacteria. Okay, then the depending upon the whether the flagella is present or not, there are two types of the bacteria. Okay, whether the flagella is present or not, there are two types of the bacteria. When the flagella it is entirely absent, then the type of the bacteria it is called as the atrichous type of bacteria. Okay, when the flagella is entirely absent, the bacterial cell it is said to be the atrichous, and when the flagella is present, then the that these type of the bacteria are called as the trichous type of the bacteria. Here you can see the different types of the trichous bacteria. Okay, two types of the bacteria depending upon the flagella: atrichous bacteria and the trichous bacteria. In the atrichous bacteria, the flagella is completely absent, while when the flagella is present, these type of the bacteria, these are said to be the trichous type of the bacteria. Okay, here in the trichous type of the bacteria, the flagella, that is their number. Their number here you can see they may be present singly, they may be present in groups, they may be present at one end, or they may be present at the two poles of the bacterial cell, or they may be present on the entire surface of the bacterial cell. And that's why, depending upon their number, their position, and their arrangement, the there are various types. There are two types of the bacteria. Okay, depending upon the number, position, and arrangement, there are two types of the bacteria. The first one, it is the non-polar. Here you can see the non-polar bacteria, and remaining these four, these are the polar bacteria. Okay, non-polar here, and here you can see the polar bacteria. Okay, polar bacteria. So first of all, we are going to discuss about the polar bacteria. Polar bacteria means what? The flagella, single or many flagella, they may be present at one or both the ends of the bacterial cell, and such type of the bacterial cell it is said to be the polar bacteria. Okay, such type of the cell it is said to be the polar bacteria. Here the first one, here you can see the monotrichous. Okay, monotrichous means, mono means single, that means the single flagella it is present at one of the pole, then it is said to be the monotrichous bacteria. Okay, when the single flagella is present at one of the pole of the bacterial cell, then it is said to be the monotrichous. Okay, here you can see the amphitrichus. Second type of the bacteria, it is the amphi. Amphi means for both the side. Here you can see the single flagellum. It is present at both the ends of the bacterial cell, and that condition it is said to be the amphitrichus. Okay, one or more flagella, these are present at both the end, and that is said to be the amphitrichus. Okay, then in the middle you can see the cephalotrichus. Cephalotrichus, when the two or more flagella these are present at both the end, then it is called as the cephalotrichus. Okay, when two or more flagella these are present at both the ends, then these are called as the cephalotrichus bacteria. Okay, and here you can see when the tuft of flagella, when the tuft or the large number of the flagella these are present at one end only, these are said to be the lophotrichus bacteria. Okay, tuft of flagella, when these are present, these are said to be the lophotrichus bacteria. Okay, that is all about the polar type of the flagellation. Depending in the polar flagellation, there are four types: monotrichous, cephalotrichous, then amphitrichous, and the lophotrichous. Okay, and here you can see the non-polar bacteria. Okay, non-polar. Non-polar, it is also referred as the peritrichus. Non-polar or the peritrichus. Here in the photograph you can see the flagella. These are not present at the polar region. Not only at the polar region, but the flagella, these are present on the entire surface of the bacterial cell. Okay, when the number of flagella, these are present on the entire surface of the bacterial cell, it is said to be the peritrichous type of the bacteria. Okay, that is about the flagella. Okay, depending upon the flagella, there are two types of the bacteria: trichous bacteria and the atrichous bacteria. Atrichous bacteria, it is also divided into the two groups, the depending upon the number, position and arrangement of the flagella. These two groups are: first one is the polar bacteria, and another one it is the non-polar or the peritrichous bacteria. Then the polar bacteria, these are then again divided into the four groups: monotrichous bacteria, cephalotrichous bacteria, amphitrichous bacteria and the lophotrichous bacteria. Non-polar type of the bacteria, these are also called as the peritrichous type of the bacteria. Okay, these flagella, these are made of a specialized protein. These are made up of specialized protein called as the flagellin. Okay, specialized protein from which the flagella is formed, it is called as the flagellin. Okay, and with the help of these flagella, the bacterial movement takes place. Okay, the movement of the bacteria, it is brought about in a clockwise or anti-clockwise manner. Okay, the movement of bacteria, it is brought about in clockwise or anti-clockwise manner. Okay, that is all about the flagella. In here, the another type of the appendages which are present in the flagella, these are said to be the pilus. Here you can see the pilus, in the singular form, it is said to be the pilus. And when there are large number of the pilus are present, it is said to be the pili. Okay, pili. These pilus, these are also called as the fimbri. Fimbriae. Okay, these pilus, these are also called as the fimbri. Okay, these pilus or the fimbri, that is, these are the minute, rigid, cylindrical appendages. These are minute, rigid, cylindrical appendages which are extending outward to the cell wall. Okay, these are extending outward to the cell wall. Then what is the difference between the pilus and the flagella? The pilus, these are smaller in size than that of the flagellum. Okay, the pilus or the fimbri, these are smaller than the flagella. Then the several hundreds of the pili or the fimbri, these are present on the surface of the bacterial cell. Okay, several hundred of the pili or the fimbri, these are present on the bacterial cell. Then these pili or the fimbri, these are made up of specialized protein called as the pilin, 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 or the fimbrilin, fimbrilin. Okay, these are made up of specialized protein called as the pilin or the fimbrilin. And pila, these are completely different from the flagella. Flagella, these are the locomotory organ, but the pilus, these are not the locomotory organ. These are not meant for the locomotion. Instead of locomotion, the pilus, that is, they confers the adhesiveness means what? With the help of these pilus, the bacterial cell get attached to the another bacterial cell. Okay, it confirms the adhesiveness. With the help of the pilus, the bacterial cell get attached with the another bacterial cell. Then when the one bacterial cell get attached to the another bacterial cell with the help of the pilus, there are certain specialized pila or the fimbri which act as a sex tube. Which act as a sex tube. Sex tube means what? The genetic material of one bacterial cell, it is transferred through this pilus into the another bacterial cell. And that's why these specialized pila, these are called as the sex pili. Okay, these are called as the sex pili. I'm repeating this one. This pilus, it is confers the adhesiveness. That means with the help of these pilus, the one bacterial cell get attached with the another bacterial cell. And when it get attached with the another bacteria, some of the pili, they will, these are specialized, and these are acting as a sex tube. That means through this tube, the genetic material of one bacterial cell, it is transferred into the another bacterial cell. And that's why these specialized pila are called as the pilus. Okay, that is all about the ultrastructure of the bacteria. Okay, the detail, the how exactly the genetic material in one bacterial cell, it is transferred in another bacterial cell through the pila, which are called as the sex pila, we will discuss in detail in the genetic recombination or the sexual reproduction of the bacteria. That is all about the ultrastructure of the bacteria.